Hello everyone, myself Khushi Chaurasiya, volunteer of Shivi Forensics. In this video, we will be discussing about some of the most important and most expected MCQs from forensic biology. Firstly, what is forensic biology? Forensic biology is a branch of forensic science that applies biological principles and techniques to analyze biological evidence such as blood, saliva, semen, hair, tissues, and DNA to help investigate crimes and identify individuals. Our first question is, which chromosome is used for male specific DNA profiling? Option A, X chromosome. Option B, Y chromosome. Option C, chromosome 21. D, mitochondrial DNA. Answer is B, Y chromosome. Explanation: the Y chromosome is present only in males and is passed from father to son. Y-STR analysis targets specific regions on the Y chromosome, making it useful in identifying the male contributor in mixed samples. It is especially important in sexual assault cases where female DNA may dominate. Our next question is, the statistical evolution of DNA evidence relies on option A, Mendelian genetics, B, population genetics, C, molecular biology, D, cytology. Answer is B, population genetics. Explanation: population genetics studies how genetic variations are distributed within a population. In forensic biology, it is used to calculate the probability of a DNA match. This helps determine how rare or common a DNA profile is in a given population. Our next question is, a false positive blood test may occur due to the presence of option A, hemoglobin, B, chlorophyll, C, antibodies, D, DNA. Answer is B, chlorophyll. Explanation: some presumptive blood tests react with substances that have peroxidase-like activity. Plant materials containing chlorophyll can trigger this reaction and give a false positive result. Therefore, confirmatory tests are needed to ensure the stain is actually blood. Our next question is, DNA profiling is least useful in option A, identifying suspects, B, paternity disputes, C, identical twins, D, mass disaster victim identification. Answer is C, identical twins. Explanations: identical twin originate from the same fertilized egg and therefore have identical nuclear DNA. Because their DNA profiles match completely, forensic DNA profiling cannot distinguish between them. This makes DNA evidence least useful when identical twins are involved. Our next question is, the species origin of blood is determined by antigen antibody reaction in option A, Kastle-Meyer test, B, Benzidine test, C, Precipitin test, D, Takayama test. Answer is C, Precipitin test. Explanations: the Precipitin test is based on a specific antigen antibody reaction. Human body protein act as antigens and react with anti-human serum. If a visible precipitate forms, it confirms that the blood is of human origin. Our next question is, which technique amplifies DNA? Option A, electrophoresis, B, PCR, C, ELISA, D, chromatography. Our answer is B, PCR. Explanation: PCR, polymerase chain reaction, is a technique used to amplify small amounts of DNA. It produces millions of copies of a specific DNA sequence. This makes it possible to analyze DNA even from very tiny or degraded forensic samples. Our next question is, which biological evidence is most useful in old skeletal remains? Option A, blood, B, soft tissues, C, nuclear DNA, D, mitochondrial DNA. Answer is D, mitochondrial DNA. Explanation: mitochondrial DNA is present in large numbers within each cell. It is more resistant to degradation than nuclear DNA. Therefore, mitochondrial DNA is especially useful for identifying old skeletal remains, bones, and teeth. Our next question is, the presence of sperm heads without tails suggests option A, old semen sample, B, agiospermia, C, vasectomy, D, erectile dysfunction. Answer is C, vasectomy. Explanations: after a vasectomy, the pathway of sperm tails to enter semen is blocked. As a result, Semen may contain only sperm heads without tails. This finding is important in forensic examinations to indicate a vasectomized male. Our next question is DNA fingerprinting was developed by option A Mendel, B Alex Jeffrey, C Watson, D Crick. Answer is B Alex Jeffrey. Explanation: Sir Alex Jeffrey developed DNA fingerprinting in 1984. Our next question is saliva contains which important enzyme? Option A trypsin, B pepsin, C amylase, D lipase. Answer is C amylase. Explanation: Saliva contains the enzyme amylase, also known as betaline. It helps in the digestion of starch by breaking it down into simpler sugars. In forensic biology, salivary amylase is important for the identification of saliva stains. Thank you everyone for watching this video.